Hi, good day everyone. Welcome to genetics class. This is for our session 2. What is genetics? It is a science that deals with heredity and variation. It is concerned with the mechanisms by which traits or characteristics are passed from parents to offspring. This picture shows a typical Filipino family. The characteristics of the parents are passed to their offspring. We know that the father of inheritance is Gregor Mendel. However, the term genetics is coined by one scientist named William Bateson. He is a British zoologist and evolutionist. The hereditary principles came about when Gregor Mendel worked on his experiments. He used this microscope during his study on the pea plants. Before Mendel, many misconceptions clouded people's thinking about heredity. Two of the prevailing errors were particularly misleading. The first was the one parent contributes most to an offspring's inherited features. Nicholas Hartsocker, one of the earliest microscopists, contended in 1964 that it was the male by way of a fully formed homunculus inside the sperm. Another deceptive notion was the concept of blended inheritance, the idea that parental traits become mixed and forever change in the offspring, as when blue and yellow pigment merge to green on a painter's palette. The picture shows the controlled process done by Gregor Mendel in his experiment. In letter A, it shows the pea plant. Its scientific name is Pisum sativum. Figure B shows the pea flower anatomy, wherein pollen is produced in the anther. Mature pollen lands on the stigma, which is connected to the ovary, becoming the pea pod. After landing, the pollen grows a tube that extends through the stigma to one of the ovules, allowing fertilization to take place. To prevent a self-fertilization, Mendel used cross-pollination. Breeders remove the anthers from the female parents before the plant produces mature pollen. Pollen is then transferred with the use of a paintbrush from the anthers of the male parent to the stigma of the female parent. Each fertilized ovule becomes an individual pea that can grow into a new pea plant. All of the peas produced from one flower are encased in the same pea pod, but these peas form from the different pollen grains, and matings within such pure breeding lines produce offspring carrying specific parental traits that remain constant from generation to generation. In Mendel's experiment, he did not only use pure breeding stocks for the member of a pair, but he also cross-fertilized pairs of plants to produce hybrids, which are the offspring of genetically dissimilar parents for each pair of antagonistic traits. The mating of parents with antagonistic traits produces hybrids. Note that each of the hybrids for the 7 antagonistic traits studied by Mendel resembles only one of the parents. The parental trait that shows up in the hybrid is known as the dominant trait. Mendel planted pure breeding green peas and pure breeding yellow peas and allowed them to grow into the parental generation or the pea generation. Cross-pollination of pure breeding parental plants produce the F1 hybrids, all of which resemble one of the parents. Self-pollination of F1 plants gives rise to an F2 generation with a 3:1 ratio of individuals resembling the two original parental types. These yellow peas, progeny of the P generation, were the beginning of what we now call the first filial generation. Mendel then harvested and counted the peas of the resulting second filial generation, the progeny of the F1 generation. The trait that appeared in all of the first filial generation, or the F1 hybrids, are yellow seeds, which means that this characteristic is dominant. And the antagonistic green pea trait that remained hidden in the F1 hybrids but reappeared in the F2 generation, or the second filial generation, is considered as recessive. To account his observation, Mendel proposed that for each trait, every plant carries two copies of a unit of inheritance, receiving one from its maternal parent and the other from the paternal parent. Today, we call these units of inheritance genes. Each unit determines the appearance of a specific characteristic. Mendel further proposed that each gene comes in alternative forms, and combination of these alternative forms determine the contrasting characteristics he was studying. Today, we call the alternative forms of a single gene alleles. The gene for pea color, for example, has yellow and green alleles. The gene for pea shape has round and wrinkled alleles. Individuals having two different alleles for a single trait are monohybrids. By the way, in his picture, this involves hybrids for a single trait, which we now call as monohybrid crosses. Why is it that Mendel used the pea plants? There are characteristics of useful experimental organisms that must be considered. One is variation. Good experimental organisms should have a number of differences detected. And recombination. Sexual reproduction, which involves gametes or sex cells from two different parents combining to form fertilized egg or zygote. This permits recombination. Controlled matings. Matings of organisms should be controlled. Parental lines should be chosen, and careful records of organisms through generations should be kept. And short life cycle. Chosen organisms would have short life cycle. For example, is the fruit fly, which is much often used in genetics because it can provide a generation of 10 days. And large number of offspring. A large number of offspring or progeny per mating is necessary to help detect the occurrence of rare events. Convenience of handling. It should be possible to raise and maintain the organism conveniently and inexpensively. Bacteria is a very useful experiment organism in this respect. Let's take a look on Mendelian laws of heredity. Where in the cell are Mendel's genes? The answer came in 1910 when Thomas Morgan at Columbia University in New York demonstrated that Mendel's genes are located on chromosomes. He proved the chromosome theory of inheritance. There are two laws of heredity by Mendel. One is law of segregation and law of independent assortment. Before we move on to the two laws of Mendel, we will tackle first the Punnett square. It shows a simple way of visualizing the results of the segregation and random union of alleles during gamete formation and fertilization. For example, the alleles Y for yellow or R for brown. The dominant alleles are represented by two capital letters for yellow. For the recessive alleles, a lowercase letter y for green. The law of segregation states that two alleles for each trait separate or segregate during gamete formation and then unite at random, one from each parent at fertilization. Figure A shows the two alleles for each trait separate during gamete formation. The same thing happened for the green pea, represented by recessive traits or lowercase letters. During gamete formation, the alleles separated or segregated into two. In figure B, the two gametes, one from each parent, unite at random at fertilization. So these are the gametes, one pollen grain and one egg, and randomly fertilize. And this is the resulting product, which is a yellow pea, a dominant trait. The law of independent assortment states that during gamete formation, different pairs of alleles segregate independently of each other. Alleles in the parental cell independently assort each other, forming possible allele combinations. That's it for this session. Stay safe and God bless. See you in our next one. Bye!